Okay, thank you very much. I'm impressed this goes down low enough for a short person. I'd like to thank the organizers for this event. It's a phenomenal event, and I spent a lot of time talking to communities. This is a fantastic showing, as will tomorrow's session be. I'd also like to say thank you to the medical professionals here for tolerating 15 minutes of science. I'll try not to make it too painful. So what I'm going to be talking about is, after a quick introduction to the problem, which I think everyone here knows only too well, I'm going to pick out aspects of recent research that touch on the issue of prevention, that touch on the issue of early treatment, and touch on diagnostics. And I'm doing this in the hope that we can find a way to incorporate science into a path forward for communities and individuals in those communities. So the problem, which I think we know, is that there are ticks, and there is Borrelia, the Lyme disease bacteria. Now, if the two of them stayed apart, or if they weren't there together at all, we wouldn't have a problem. But deer are a very good way of making ticks. Mice are a very good way for the bacteria to overwinter. Ticks do eventually get killed off in the cold. So the bacteria, their version of Florida, is a nice mouse. And the problem is that when you have an infected tick, because it fed on a mouse, that tick can find its way onto our pets, it can find its way onto people, and then you have the potential for disease transmission. And you get relatively disturbing situations like this, which was a picture sent to me from Lunenburg. Okay, so we did mention briefly climate change. Yes, this is an aspect of climate change. I should mention that I'm based in New Brunswick, so most of my examples will be New Brunswick. We can assume that it's the same species of people in both provinces, so it's applicable. In terms of the climate, New Brunswick is more or less on the advancing edge of tick moving in. They're establishing very nicely their endemic populations through the southern half-ish of the province and up the coast. In 20 years' time, that's what it's expected to be like. In 50 years' time, that's what we're expecting. And congratulations, Nova Scotia, you're already there. Ahead of the curve, as always. Okay, so as I said, I work in New Brunswick. I spend a lot of time working on the ticks there. People send me ticks. I squish them up, take their DNA, because it makes me happy. Everyone needs a hobby. We don't solicit ticks from Nova Scotia, largely because I don't have funding to work on Nova Scotia ticks, but often enough, it's just easier to test Nova Scotian ticks than send them back with an explanation of, sorry, wrong province. So over the past 5 years, we've tested over 700 ticks from Nova Scotia, and these are the results we've seen. In terms of the tick species, about 60% have been black-legged ticks. What that means is that 40% have been primarily the American dog tick, Dermacentor variabilis. And from a human health perspective, that means that 40% of the people who are panicking because they found a tick on them have actually got a tick that isn't a Lyme disease vector. Now, the dog ticks do transmit other diseases, but much more rarely. So one of the first interventions that could be done is to simply figure out what kind of tick that is on a person. For the people who have black-legged ticks on them, these are our infection results. This is for Cumberland County, so northern Nova Scotia, and we're looking at roughly 20-ish% infection frequency. Dr. Cran mentioned that there's data for the rest of Nova Scotia. I admit I haven't done an exhaustive search, but people do ask me about that, so it'd be great to see the rest of Nova Scotia data. I know Pictou County is about another 5%. This area, I'd expect to be double that. All this information is publicly available if you want to poke at it. It's what we call an interactive map, which is mostly meaningless. So with that as a background, the idea here is that from a biologist's point of view, the ticks are here. They're not going to go anywhere, and in fact, they like it here. They've moved to the Maritimes, they love it. And short of dropping a nuclear bomb on the region, they're not going anywhere. So we have to get used to them. And where there are ticks, they will interact with humans. So we have to peel people off this pathway leading to tick vector diseases, and there are 3 ways to be peeled off. One is prevention, basically stopping the ticks from biting in the first place. If that doesn't work, and prevention is never 100%, we can at least figure out there's a tick, catch the tick early and treat appropriately, and if that doesn't work, treat later disease. All of those pathways are ways of stopping people from getting to the end of the pathway and falling off. So to break that down and to introduce recent scientific stuff that can help, in terms of prevention, there's surveillance and there's prevention practices. I'm going to touch on both. For early detection, 2 important components: what kind of tick is it, and is it infected? So tick ID and tick testing, and ideally treatment in response to that information. To deal with the later disease, there's the question of diagnostics. That's been mentioned previously, which is great. There's treatment, which I'm not a physician, so that's your problem, not mine. Excellent. So to talk about surveillance, 2 components: surveillance. We can do tick surveillance, canine surveillance, or human surveillance. I don't think anyone will be surprised to know that I'm mentioning the work I did with Dr. Hawkins on this. This is looking at how good our human surveillance information is. And it turns out that we're under-detecting Lyme disease is not controversial. That's true everywhere in the world. To some extent, that's intrinsic to surveillance systems. But you do need to know to what extent it's been under-detected. We estimated we're detecting 10-ish% of the cases. Public Health Agency of Canada estimated detecting 30% of the cases. That's actually fine. What scientists love to do is argue about numbers, so I consider this a great success that we've moved to the point where we're arguing about numbers. It keeps us busy, keeps scientists out of trouble. It's all good. The concern here is in terms of when we look at the data, everyone keeps saying, well, there are 2,000 people with Lyme disease in Canada. Are there? First of all, if we're not counting well, we won't know how bad the problem is. And secondly, for the people who aren't counted, are they being treated and not counted, or are they not being counted because they're not being treated? And that's the concerning part here. That's it for surveillance, because I don't have much time. I'm going to mention prevention. We have talks tomorrow by Dr. Rob Murray on prevention, which is fantastic. So I'm going to pick out one aspect on prevention, but I will make a nod to the Tick Encounter Resource Center at the University of Rhode Island. It's a huge number of resources that are very useful for all age groups and very community-centered things you can do. So that's worth having a look at. I've also stolen their tick engorgement chart. And the reason I've done that is because we've talked a number of times this morning about the importance of having science-based, evidence-based messaging. And one of the aspects that come up is how long is the safe period for the ticks? And if you look at, these are animal studies, so if you look at the studies where people have plunked a tick on some sort of rodent and checked how long it takes to transmit, there are cases where it transmits under 24 hours. And it's really important that the messaging that goes out reflects what people in the community are experiencing. And I hear a lot of this. I'm sure the tick wasn't on me that long, and yet I got sick. I'm also worried whenever I see graphs such as the one you presented, Dr. Hatchett, showing the nice nothing happening before 24 hours. That's a lovely depiction of an idea, but it's not data. So that with leaving that, in terms of early detection, we've got 2 components: tick ID, tick testing. So for the ticks that feed on humans, you deal primarily with 2 different species. There are other ticks that will take a human if they're desperate. The dog tick or the wood tick is the one with white on its back. It's browny orange as opposed to the black-legged tick, which is orangey orange sometimes, and it doesn't have black legs, sort of annoying. To distinguish them, the one with the white on its back is the less disturbing kind if you have to have a tick on you. So in terms of identifying them, your options are, you can work it out yourself, but if you're panicking because there's this blob of blood-sucking nastiness on your arm, You can take a picture of it. Nova Scotia Museum of Natural History has done a great job of trying to deal with the tick problem. They will still identify ticks if you email them pictures. I can do the same. There's also a service called eTick, which has a trained entomologist identify tick pictures. That's not officially available to Nova Scotia residents, although there is a workaround. For tick testing, excuse me, there are a number of options. Depending where you are in the country, there's testing through government public health agencies or the medical system. That varies on where you are. If that's not working for you, there are a variety of commercial options. I've circled one called Genetics, not just because I love the pun, but I share a lab with them, and in the interest of full disclosure, I get no money for this, but I work with them, so I know them. Has the advantage that you're paying for it in Canadian dollars, your tick doesn't get seized at the border, and they will test for co-infections. To deal with the issue of diagnostics, I'm not going to say that much on it, but I'm going to point out that the role of diagnostics is to detect what's present in the region. And that's an area where the research community really does need to step up. In the United States, it's very well understood that we have different Borrelia species. This map from the CDC puts them in different geographic regions, which is a bit overstating the case. But we have different species, and that's understood. They will have different detection probabilities with the diagnostics. For Canada, or for this region, we have some work from the Public Health Agency of Canada. For one species, Borrelia miyamotoi, which is a relapsing fever, we know it's present across the country in low numbers. A survey of the past few years in New Brunswick has picked up a fairly rapid increase in this pathogen. Another species, Borrelia basseti, we've checked that in Nova Scotia and New Brunswick. Again, fairly rare, but it is here, and it's a problem because it's been considered to have a predilection for hitting the heart. The ones I worry about quite a bit are the so-called European species. They've been detected in ticks that hang out on seabirds. We're a coastal area. Seabirds cross the ocean. They hang out on islands. Their ticks end up on mice, which then get onto the mainland. And if that gets into the wildlife, we're going to have a number of the European species here, that being a problem because Guarini is supposed to have fairly strong neurological effects. Final point on this, before I try to persuade you that ticks are actually interesting and worth studying, is to point out that ticks are essentially walking crawly bags of pathogens. So it's very rare that a tick will have just one pathogen in it, just Lyme disease. It's the most common. But this is a study of the so-called, the Tickplex, that's its brand name. There's a similar product in the States called the Serochip. What it does is it tests for your antibody response to a number of pathogens. And the second rather cryptic bar up there shows the number of cases where people were reactive only to Borrelia. The longer bars show that more people, vastly more people, were reactive to a number of other tick-borne pathogens in addition to Borrelia. And we're not going to go into that. In terms of the diagnostics as such, the serological diagnostics, the ELISA and the Western blot, really have underpinned a lot of medical diagnostics for a very long time. These are very well understood diagnostics. They're also fairly old. The ELISA's will celebrate its 50th birthday in 2 years, so it's not quite as old as I am. However, there are other things out there. People have talked about the ELISpot in terms of sensitivity, specificity. There are some newer techniques coming along. The one that I find really promising is using Borrelia viruses to find out if someone's got a Borrelia infection. For every Borrelia species, it has its own suite of viruses. They're present in hundreds to thousands more copies than the Borrelia, and it's transmitted in the blood, so they can actually be picked out quite easily by PCR from the blood. Clinical tests have been very successful in Europe, and it's commercially available there. So there are options there for better diagnostics, and that's something where the research community could actually contribute. My last point is, this is the slide where I usually try to persuade my students that tick spit is exciting. Sometimes that's a stretch, so I'm not going to try to persuade you that tick spit is exciting. But I will point out in this somewhat garish slide that tick saliva contains a lot of proteins and a lot of other active compounds. Some of them are there to help the tick. The anticoagulant is there so that it can keep slurping your blood for a good week. The anesthetic is there so you don't notice while it has a snack on you. But the other components are there because the bacteria in the tick have changed the tick's genome and changed its gene expression to make the tick a perfect tool for them to get into your bloodstream. So we're looking at millions of years of evolution where the bacteria have honed the tick to be a delivery vehicle. It's complicated, which means a simple answer is probably not going to be there. So to recap, the ideal situation is where we prune people off the pathway to tick-borne disease at any one of the early stages. The non-ideal situation is where we don't do that. In words, in terms of prevention, there are many, many opportunities for disease reduction. Prevention messages work best if they're grounded in community experience and also grounded in good science. And unfortunately, prevention, while very powerful, will never be completely protective. In terms of early Lyme disease, I can say this blithely, being in a province next door, but province-wide tick ID would be tremendously helpful. Province-wide tick testing would be very useful, and coupling the results of tick testing to treatment would be even better because it would get around the whole issue of treating people unnecessarily because they had a tick on them. At least you know that if you're treating them, it's for a reason. Finally, in terms of diagnostics, we need to make sure the diagnostics detect what's here, which means we'd have to know what's actually here. And it would be good to include things other than Borrelia. And the last point is simply that it's tricky. We do live in a beautiful area. I still garden, I still hike, but please do your tick checks. That's it for me.
She says questions are straight to Ralph, but I think we will take an opportunity for questions. Sorry. I like your humor. I want to hear what you're going to say. Do we have any questions for Dr. Lloyd?
Right here.
Right over there.
Oh, we want to talk to Ralph.
Dr. Lloyd, you really make science sound fun.
So my
you
students,
said,
please.
You said the tick was like the optimum delivery vehicle. You spend a lot of time with them. Do you know why the ticks, and are you seeing other insects as evolving to be carriers in this way?
A lot of insects, particularly the blood-feeding insects, they have evolved to take our blood, and they have evolved to transmit pathogens. Is that relevant in terms of Lyme disease? Let's just focus on the ticks for now. But other pathogens, yeah, I mean, watch out for mosquitoes too. Fortunately, your bug repellent will look after them because mosquitoes transmit stuff too. That's a terrible answer, sorry.
Yes, thank you very much. Very interesting presentation. In terms of sending ticks in to be identified, is there a time period when it's too late, or does it have to be done immediately when you find the tick?
For both identification and testing of the tick to see if it was infected, identification is morphological, but if you freeze the tick or just let it dry out, as long as you don't hit it with a hammer, we could usually manage. Yesterday I got a tick leg. That was a challenge. For the tick testing, we can take pretty much anything, dried, frozen, mashed. We've even done covered in ketchup, and please don't ask me why.
Come on, more questions. Her answers are great.
Is there any possibility that we are receiving Lyme disease through through eating deer meat?
Deer meat should not be a likely source of infection, and there are a couple reasons for that. One is that the deer are actually very good at clearing the Lyme disease infection. So deer are a great way of making more ticks. As are other large animals full of blood, there's nothing terribly special about deer. So deer are important in making more ticks, but they don't carry the Lyme disease bacteria except very transiently. You might have a risk, well, if you got a deer in that transient phase, but I'm assuming that you're not eating it rare, or raw. If you are, there are many other things to worry about, so probably not.
Question over here, Vett. Hi. In terms of post-Lyme disease syndrome, I know that there's a couple of monkey studies where they treated monkeys were infected with Lyme, they treated them with antibiotics, then they sacrificed the monkeys, did a post-mortem analysis of some of their tissues, found some viable spirochetes in some of those tissues, and then if memory serves, I think it was either you or one of the students in your lab that presented at the event at Dow last year about, I think it was a human post-mortem study as well, where you found spirochetes. And I'm just wondering if you could comment on that, as any insight you might have, whether there might be some information giving us more insight into what's happening with the post-Lyme disease syndrome.
Okay. So I think, as with most diseases, and I'm speaking out of my area of expertise here because I am a biologist, but in a general sense, it's going to be complex and there are going to be many causes. But speaking specifically to whether Borrelia can survive standard treatment, certainly the Embers monkey study showed they gave the monkey thorough treatment and still recovered the bacteria. The same is true in a number of human studies. Living bacteria have been identified by culture. There are a number of post-mortem studies that have detected Borrelia bacteria post-mortem after standard treatment. Obviously, at that point, you don't know if you're detecting live bacteria or not because it's from a deceased body. And there's one study which I thought was particularly interesting. They used zoodiagnosis, so they treated a person and then put uninfected ticks on them. And the idea was that if there was living bacteria, it would end up in the tick again. And they did find that in a limited number of cases, but they did find that. So there certainly does seem to be evidence that the bacteria can persist after a short-term treatment.
Thank you.
I don't know if there are any pathologists in the room, but I was wondering if a live punch biopsy of rash could you identify what's there if you did?
I can't help you with that question.
I keep trying to escape. If the bacteria are present, there have been a number of studies that show that in early infection, they have been able to culture Borrelia and also isolate its DNA from skin punches. Later disease, I think it would be much more challenging because the bacteria have disseminated through the body, and the body's a big place, and there are not that many bacteria. So it's possible. I'm going to give you the answer of any scientist, which is we'd have to try.